Start.
Okay, thank you, Sharon. So, and thank you, Laura, for being able to join and share your important data and vision on the industry with us. So, it's my great pleasure to introduce Laura. Laura Gusco is a full professor at the Department of Agriculture, Forest, and Food Sciences at the University of Turin. She focused on fish, rabbit, and poultry farming and nutrition, assessing performance and the impact of alternative ingredients on protein quality. Now, since 2012, her research focused on the use of insect meal as innovative ingredient for animal nutrition and the optimization of insect rearing for the production of high-quality raw materials, focusing on the black soldier fly and the mealworm. And she also involves in many projects related to insect feed. Beside that, Laura is the president of the study commission insect in the EAAP, and also section editor at the journal Animal, and a member of the editorial board of the Journal of Insect as Food and Feed. So thanks again, Laura, and I hope that it will be as fascinating as usual. And let's start.
Okay, thank you very much, Eden, for your presentation, and thanks to the 27 people. I don't see 20 or 30 people that are online. 30 minus 3, Eden, myself, and you. So 27 people that are online. So thanks for giving me this opportunity. I will try to share my screen. Let's see if I succeed with this. Okay. I'm trying to share from the video. So I will share this one and then this one. Just let me know if you see one slide. Is it okay?
No, you're not sharing anything.
Oh, why? Okay, let's try to do it again on the video. What about now?
Now we see your Gmail.
Still my Gmail.
Now it's good.
Okay,
Perfect.
now it's good. You see the screen. Okay, so welcome everybody. Well, I'm really happy to be here, and thanks a lot to Friesen for this very nice invitation and to give me the opportunity to tell you something about black soldier fly and insects in general. So when I discuss with Eden about what to present, I asked him which is the audience I have to expect. And he told me participants in this seminar come from various backgrounds in the field and work with black soldier fly, genetics, nutrition, processing, fast utilization, etc. So nearly everything. So you know already nearly everything about the insects and the black soldier fly. So my task is not easy. So I decided to try to present you some topics that I think are important for the research now, and that could be also important in the future for the development of the sector and to help the development of the sector. So why insects in animal feed? Well, we all know that the global feed production has increased in the last years. If you look at from 2012 to now, we have a plus 37%, and this growth has been continuous and is expected to continue because of the growing population and the growing need and request for animal products. So the feed sector need new sustainable ingredients, and of course, insects are considered as an emerging and sustainable source. Well, of course, insects are rich in nutrients. They contain proteins, they contain valuable amino acids. Remember that insects are animals, and being animals, the essential amino acid content is better than the one in plant protein sources. They contain lipids, they contain vitamins, they contain minerals. And well, from this slide, you can see that, of course, insects are a very interesting tool for the circular economy, because from waste, from organic waste, you can grow insects, you can produce different kind of products, you can produce live larvae, you can produce processed animal proteins, you can produce oil that can be used for animal feed purposes. You can produce frass that can be used for agricultural purposes, and then back again to the soil, and then back again to organic waste, and the circle starts again. And as you can see, you can also reach and match several sustainable development goals. But insects are not only a source of nutrients. I know that you read a lot about insects as nutrient sources, so I will try to focus more on what could be the side effect of insects. So the first side effect is the bioactive compounds that insects contain. You know that insects contain some specific compounds such as antimicrobial peptide, chitin, fatty acid that can act as immune stimulation, that can have some microbiota modulation effect, that can have some antimicrobial effect. And in a moment, in an era where we are facing a huge problem with the antimicrobial resistance, it's quite important and quite nice to have some natural biomolecules that are able to help facing this kind of resistance, because normally we don't have anymore many instruments when we are in a poultry farm or in a rabbit farm. We don't have many antibiotics or
Laura, you are muted.
But was I mute since the beginning?
No, just
someone mute me. Okay, so I was just, why my slides are not going on anymore. I'm working with two screens. Okay, do you see now? Okay, so I was saying that it's important to have some antimicrobial substance trying to face what are the bigger loss in animal farms, because of disease. It's a big problem, and insects can have this kind of bioactive compound. So let's start with the first one, which is antimicrobial peptide. What are antimicrobial peptide? They are small protein that exhibit action against some bacteria, fungi, parasite, virus, and they can act in different ways. Two mechanisms, mainly the first one is they can kill microbes by interacting with the membranes. They used to produce a kind of problems with the membrane cell, so the membrane cell go towards a rupture. Or they also can inhibit the function of the intracellular function, so that results in the death of the bacteria. And the second way of action is that they can stimulate the immune system of the host, so of the poultry or of the fish, in different ways. And why this happens? Because insects are able to produce many kind of antimicrobial peptide when they are in some way infested by bacteria or by parasite. They will react, so they will defend themselves toward those bacteria or parasites. So what's happened is that insects have two main mechanisms to produce antimicrobial peptide. The first one is the fat body, which is the equivalent of the liver. And then they produce antimicrobial peptide, and they release those antimicrobial peptide into the hemolymph. And this is the main mechanism of production. But then, as it happens also in vertebrates, insects are able to produce antimicrobial peptides through the epithelial cells. And when there is, for example, a parasite or a fungi, they can produce antimicrobial peptide. And the results is just to boost the immune system of those insects. And if you think about sometimes the harsh environment where insects are living, it is clear that this production is really important for them. And the antimicrobial peptide that insects are able to release are active against different kind of gram-positive, gram-negative bacteria, fungi, etc. And you can find, of course, some review on that. Just to give you some examples to demonstrate the effectiveness of antimicrobial peptide of the black soda fly here, what Park and all did, they just activate the production of antimicrobial peptide in a larvae of black soda fly, spiking them with a needle. So they freeze-dry the larvae, they homogenize the larvae, and then they extract the peptide from the larvae. And they tested those peptides against a broad spectrum of bacteria, and they found some very interesting antibacterial activity, and again towards a positive, gram-positive or gram-negative bacteria. But what I would like to highlight here is that they found an action against methicillin-resistant Staphylococcus aureus, which is one of the bigger threats that also human health is facing, because if you contract a methicillin-resistant Staphylococcus aureus when you are in the hospital, then you are in trouble because there is no antibiotic that is working against this. So this is something really, really interesting. Another research that has been done here is that they tested the performance of black soda fly against Escherichia coli and Salmonella enterica. So what they did is that they contaminate chicken manure with Escherichia coli and Salmonella. Then they inoculate the manure with 11-dose black soda fly larvae, and they incubate those larvae at different temperatures, 23, 27, and 32 for different days. And what they found is that they found a very strong decrease of Escherichia coli, in particular in larvae in manure that were incubated with larvae at 27 and 32 degrees for 3 days. And this was very evident against the manure that was incubated at 23 degrees. So this proves the effectiveness of larvae against those bacteria, and also the fact that larvae are active at 27 and 32 degrees if compared to 23, which is quite normal, because if you look at larvae at 23 degrees, clearly black soda fly is living at 23 degrees, but the metabolism is not that active at 27 or 32. So another, well, what about the action of chitin? So chitin, everybody knows what chitin is. Chitin is it's a polysaccharide, structural polysaccharide that compose the exoskeleton of crustacean, of insects, the membranes and the cells of vegetable, of plants, of algae. And chitin from crustacean has been seen to have a very strong action, different action, anti-inflammatory, antibacterial, immunostimulant, anticoagulant. So, and it is already used in application for medicine, for pharmaceutical. And then the question is, what about the insect chitin? So is the insect, has the insect chitin the same effect than the crustacean one? We know that there are some differences between the chitin from insect and the chitin from crustacean in particles, in structure, in composition. So usually the one that has the highest anti-inflammatory effect are the small particles. Then the chitin structure can be alpha, beta, gamma, and also the composition is different between crustacean and insects. So the crustacean one is a matrix of protein and mineral, while the chitin of insect is a matrix of protein, lipids, and other compounds. So there could be some differences, but more and more evidence show that the chitin also from insects can have positive impact on the animal health. So some examples here, trials with yellow catfish, 4 levels for 4 diets, and they use Tenebrio molitor, 0, 9, 18, and 27%. And as you can see, in fish that were fed 18% of Tenebrio molitor meal, they found some nice results as far as antioxidants parameters are involved, so malondoidal content and superoxidant dismutase activity. They found an increase in the parameters of the immune system, so the lysozyme activity and the immunoglobulin levels, as well as an upregulation of some genes that are related to the immune system. And what they also did is that they challenged yellow catfish with Ergoacelia ictaluri, which is an issue in yellow catfish farms. And they found a really strong resistance and a survival rate that was definitely better in the fish-fed insect compared to the fish-fed the control diet. Another example on immunostimulation, still with yellow catfish, but in that case using black soda fly meal, 2 levels, 10 and 22%. And as you can see, at both levels, we had an increase in the immune system response. So lysozyme activity that was boosted in some way. And also, in both cases, we had some good results also for the performance with an increase in weight and also a lowest feed conversion ratio. Immunostimulation, again, but in this case in poultry, and in this case they use black soda fly meal not really as an ingredient, but more as an additive, so at low level of inclusion. And so 1, 2, and 3%. And once again, they found increase in the, well, the performance, but also in the response of the immune system. So T lymphocyte, lysozyme activity, spleen lymphocyte proliferation. And they challenged the birds with Salmonella gallinarum, and they found a survival rate that was increased. So they conclude that black soda fly larva meal at low level of inclusion stimulate the non-specific immune response of the birds. Concerning the action in the microbiota modulation of the chitin, here in this research that was performed by Borrelli in laying hens, they investigate, of course, the performance, etc., but they investigate the cecal microbiota modulation, and they found a development of bacteria that have a chitin activity, a chitin degradation activity, and having a chitin degradation activity, they also relate that with an increase in the short-chain fatty acid production. And the short-chain fatty acid production is positive because it can result in a better performance of the birds. So it is like as the chitin can also have a kind of probiotic effect. And more or less the same result was also obtained with fish, in this case with redwood trout. And what they did is that they used a chitin from crustacean and also from exuviant of black soda fly. And as you can see here, they modulate the microbiota of the fish. They increase the biodiversity of the microbiota, and they increase some genera that have once again a kind of tinolytic activity and a production of short-chain fatty acid bacteria. So with a positive modulation of the gut microbiota community. And I have to say that more or less all the research that have investigated the microbiota modulation by the insects, I mean, give this kind of results. So there are more and more, evidence that proves this kind of results. And if you want to know more, you can go to look at this very nice review that was delivered in Animal Frontier, and you can download it for free in the web as it is an open access. What about fatty acids that are contained in insects? You know, at the beginning, we were looking at insects only for the protein content because we were looking for alternative protein. But then we also seen that there are not only proteins. So fatty acid and lipid profile is also very important. And when you are looking at the fatty acid profile of the insects, clearly not all the insects are the same. You have a very important impact of the insect species on the fatty acid profile. So usually we have as a saturated fatty acid, the most representative is the palmitic acid, with the exception of black soda fly, which is very rich in lauric acid, and this is a very interesting fatty acid. Then we have oleic acid, and as long as, as far as polyunsaturated fatty acid is concerned, we mainly have omega-6 fatty acids, so linoleic acid, and to a lesser extent, omega-3 fatty acid. Omega-3 fatty acid is more present in bone mixed mori, in the silkworm, but all the other insects contain more linoleic. What about the long-chain polyunsaturated fatty acid, like EPA and DHA? Well, those are very interesting from a point of view of animal nutrition and also in human nutrition, but there are no evidence in the biosynthesis of those fatty acids from the insects. But what can happen is that the insects are able to upload those long-chain polyunsaturated fatty acids from the substrate. So you can modulate the fatty acid profile of the insects with the substrate. And what I want to highlight here is, once again, is the high level of lauric acid that is present in the black soda fly larvae. And this is research we perform using oil in substitution of soybean oil in the feeding of rabbits. We did some performance, product quality, etc. But also we also investigate the impact of these oils against bacteria that are pathogen and that are a problem in the farms, in the rabbit farms, so Pastorella multocida, Yersinia enterocolitica, and Listeria monocytogenes, and look at the black soda fly oil impact, which really control and reduce the development of those bacteria. So this is a very important result. And once again, some review. Another hot topics, I think, is the insect meal digestibility. I will not describe in a very detailed this, but what I think that is still lacking in the research is the determination of the digestibility of the insect meal itself. So usually when we are doing research, including insect meal in animal feed, we also investigate the digestibility of the whole diet. But what is missing, and there are not that many research on that, is how the insect meal itself is digested by the animals. So there are research that investigate the digestibility of different insect meal, for example, taking Tenebrio and Hermetia, full fat, defatted, and it has been seen that the process, the insect species affect the digestibility of the product. And why it is important to have the value of the digestibility of the product, because it helps, it enables you to properly formulate diets for animals. Usually when you are formulating a diet for a poultry, for a fish, for a rabbit, for a pigs, you formulate the diet based on the digestibility value of the different ingredients you are including into the diet. So it's important to have this kind of value for the insects. So globally, insect meal are characterized by a good to high digestibility value. And if you look, if you compare those value against the value that are obtained for the other processed animal protein, the value that are obtained for insect are definitely very, very good. But of course, there are many different parameters that can impact. So the insect species, the composition, which is impacted by the substrate, and there is a huge impact also of the processing of the insect meal. So the way you are processing the product will impact its digestibility. And you have to consider different parameters when you are comparing the results. If you want to have some information, we deliver some very nice best practice sheets during the Smooth in Chain project. But as I told you, I think that for pigs, there are 2-3 research, 5 or maybe 4 or 5 for poultry, and 8 for fish on that. So there is still a lot of research to be done. And well, this is a topic we are going to investigate. So what about insect for feed? Well, clearly the insect meal or the insect oil in animal feed nutrition, it works. As long as you properly formulate the diet for your animals, it has been shown that it works. You know, it's an ingredient, a very valuable ingredient. You just have to formulate the diet in order to cover the requirements of your animal. See, this is why it's very important to have the value of the digestibility. And in this publication, what we did is that we made a review on the level of inclusion that were suggested. So for fish and for shrimps, around 25 to 30% of inclusion, and for pigs and broiler, around 10% of inclusion, which are also level of inclusion that are quite reasonable if we think about the quantity of insect meal that we have available in the market. But discussing about insects for feed, I think that one very interesting and one hot topic is the use of insect meal and insect-derived products in ruminant nutrition. You know that in ruminants, at least in Europe, you cannot use animal products in the feed because it's forbidden by the legislation, the feed ban, so the legislation that was released after the BSA issue. while there is no restriction for the insect fat or insect oil in ruminant nutrition. Well, in other countries, of course, other kind of regulation can apply. There are countries where there is no regulation at all, countries that authorize the insect oil but not the insect meal, or the opposite, or countries that everything is forbidden. But there are some research, not a lot, that are ongoing on this topic, and this is a review that was performed by Manuela Renna, and once again you can find the publication in Animal Frontiers. They review all the trials that were done with ruminants in using insect products. So 12 trials, 9 trials with insect meal, 3 with insect oil, 9 in vitro and 3 in vivo. It is difficult to perform in vivo trials with ruminants. It's very expensive. And then you have the European legislation, which is not positive for that. And what it is very important to see is that globally, insect meal seems to decrease the in vitro digestibility if you compare that to plant protein meal. And there are some explanations for that. Is that the first explanation is, in particular, the full fat insect meal or the insect oil will contain a high level of lipid, of ether extract, and especially the insect meal and insect oil that contain unsaturated fatty acid. The unsaturated fatty acids are toxic for the cellulotic microbiota in the rumen, and this decreases the digestibility. And there is also another explanation, but it has been, how to say, have a minor role, which is the chitin that is low or non-degradable by the rumen microbiota. And well, once again, if you can see here, you see that the results are impacted by different parameters, such the insect species, the level of processing, etc. But what I also want to highlight in my slide is that, as you can see, and maybe you know, when the protein enter into the rumen, the protein is degraded into ammonia nitrogen, which serve as a substrate for the fermentation, and then. As the protein is the most expensive part in an animal diet, what feed producers try to do is that they try to protect the protein, to have a protein bypass, in order to bypass the rumen, to so bypass this kind of degradation and go into the intestine, the small intestine, where they can be used for, production, for performance. And so in that case, it seems that the lower digestibility of the protein into the rumen could be a positive effect, as this means that they could deliver into the intestine a higher level of protein that could be used for performance. So this is interesting. So a kind of natural bypass protein. Another very interesting impact, and I think that this is something that definitely worth the pain to investigate, is that it has been shown, and you can see that there are many data that are in the same way, is that the insect products can modulate the methane emission. And as you know, in ruminants, the methane emission is one of the big issue, for the environment. And it can be, so this mechanism can be explained in 2 ways. The first one is that the saturated fatty acid, and in particular the one from black soda fly, so the lauric acid, has an antibacterial and antimethanogenic effect. And the second one is also that some fatty acids have an impact on the protozoa, so they decrease the protozoa count, the protozoa into the rumen, and those protozoa are protozoa that are producing methane. So clearly you can see that this is a very important and make very valuable the insect oil. So insect oil can of course be used as a source of energy, but I think that it is definitely more interesting to use that as an additive for this kind of effect, because it can have, of course, a positive impact on the environment, but also in animal performance, as we know that there is a very big role of the methane emission on the energy loss and then a loss for the performance of the animals. And if you want to know more about that, we have a project that is the IGA project, where we already performed the trials with ruminants, and of course we investigate all the parameters, the performance, the production of the milk, the quality of the milk with the panel test, but we also investigate the lipid bioregeneration, the methane emission, and the protozoa impact. And if you want to know more, Manuela will be very happy to give you a lot of information on that. And another very nice result that we obtained from this trial is that we had a cow that had a mastitis. So we isolate the bacteria responsible of this mastitis. It was a Staphylococcus aureus. And then we tested the black soldier fly oil against this Staphylococcus, and we had an impact, we had an effect. So this can open also the use of black soldier fly oil, maybe as a dipping solution to treat the mastitis. This is a very nice kind of sum up figure I found in a recent article, where they investigate the impact of insect meal and insect oil in feed for fish, and they found some nice impact on the palatability, on the expansion rate of the feed, and this can have an impact, a positive impact on the sustainability of the feed, on the management of the feed. They had impact with the feed intake, then improved growth. But overall, I would like to stress the positive impact on good health and on the immune stimulating effects, which can really help in decreasing the use of other kind of treatments. Another very hot topic is the insect substrate digestibility. So we know that insects are living into a substrate. So they are bioconverting the substrate. Not all the substrates are the same, and insects are not bioconverting all the substrate in the same way. So it's important to assess the digestibility of the substrate by the insects, like any kind of animal. I'm doing my digestibility trials with fish. Why not to do that with insects? The problem is that insects are living inside the substrate, so it's difficult to have the digestibility of the substrate. And there is a very, I would say, a milestone on that case for this kind of topic, which is a paper that was delivered by Guillaume that evaluate the asymptote estimate digestibility, which is the maximal fraction of feed that black soldier fly and the associated microbiota, which are living within the gut of the black soldier fly and also within the substrate, can potentially digest. And as you can see, well, they did some trials with different densities, and they were able to assess the dry matter, the starch, the ether extract, the nitrogen, the ash, and the gross energy digestibility. But of course, once again, there are a lot of research to do. This is really the first trial that has been done in that way. And I think that this is once again a very interesting topic to investigate. Another topic is to investigate which are the nutritional requirements of the insects. Well, black soldier fly, Tenebrio molitor, any kind of insect that is used, that is mass produced for feed purposes, of course, they are able to eat, I would say, nearly everything. But as any kind of animal, if you are delivering them with a proper feed, the results will be definitely better. So there are investigations on the optimal protein level, lipid level, ratio between carbs and lipids, etc., and carbs and protein. And I know that the industry is already well advanced on that sense, but also from a research point of view, you know, the role of the university is to deliver publication that give some evidence on what to do. And if you want to join, there is a cost action on that, which is by the Gene Tonic. The name is quite funny, but they are working hard. In the evening, I would say that the gene is also part of the cost action, but then you can join the cost. You know, cost action are open to everybody, and you can contact Dennis or Sarah for that. Another hot topic is genetic. So there has been a huge improvement in plant, in animal production for growth, production of the different parameters, the feed efficiency, the resistance for the disease. Look at the difference between a poultry 50 years ago and now. Okay, we can still argue if the one that we have now is still a poultry or not, but definitely this is also the results of the genetic improvement. And well, for insects, there is still a huge a huge potential that can be exploited in that way. And another cost action is on insect genetic. And I didn't put any slide on that, but there will be a workshop organized by the insect genetic cost group that will be in the last week of January in Athens. So if you want information, I can send you the information and the link. The early bird is still open until the 27th of December, if I'm correct. Welfare. Okay. Welfare is also a very hot topic. We can agree or not about the idea of discussing the welfare of insects. I don't know if Erich Katz is on the audience. I already see his hair like that.
Yes, he is.
He's okay. Hi, Erich. Yeah, clearly, we are discussing about insect welfare because it is, you know, people are really care about welfare. Welfare of animal is a huge topic for the animals, and also clearly, I see you, Erich, hi. And clearly, we cannot, well, the industry cannot do without the insect welfare. So as for the other animals, you know, the 5 freedoms for bundles were the drivers of the welfare for animals and also for insect welfare. But then insects, we are discussing about insects, but there are so many insect species. And the idea of doing only one legislation on insect welfare, it is too restrictive, in my opinion. We need to take care about all the different differences, you know, existing from an anatomy point of view, physiology point of view. They have different kind of behavior. They have different requirements. There are some insects that, what about the density of insects? You know, if you don't put all the blacks of the fly very close one to each other, they will not be happy. So, you know, we cannot put some limits in the density without knowing really the behavior of the insect. So I think it's really important to have not only one legislation on insect welfare, and this requires a lot of research. It requires time. So I really hope that we will take the time to do it properly. Otherwise, the risk is that we will have something that will be too restrictive and not applicable for all the insect species. And in that sense, I'm really sure that the collaboration between researchers and producers is really important, even if not always easy to have, because I know that, well, you know, the industry has other mechanisms that research, sometimes they are not that willing to share and to collaborate because they also have their secrets, which is normal. But then I think that this is something important to investigate. And you, of course, you have some nice publication, a lot of publication. We had a very interesting lecture at the Insect to Feed the World conference in Singapore by Megan Barrett, also on the way of killing insects. You know, also the killing of insects will have an impact on the quality of the product that we will have at the end. So, and this is very important for the application in food and feed. Another topic is frass. So I think that you know what frass is, and that there is I think a quite big interest now for the frass, because at the end frass is a big part of the process, of the whole process, and to be able to apply those frass as an amendment in the crops or the plants, etc., is a very nice issue. If you want to have the circularity and to apply everything on the what I told you from now, and this is a project, the Alpha Agromet project. So we grow insects, Tenebrio and Hermetia on bite stream, on agricultural side stream. We fed live larvae to poultry, so as a source of nutrient, but also to improve the welfare of poultry. They are very, very happy when they eat live larvae. And then we apply the frass from insects and also the poultry manure into the crop to have the circularity. 10 minutes left. Very quickly, the challenges. There are challenges. You know that I like insects. I'm really involved in this and I believe in this sector, but we cannot hide. We cannot masquerade the different challenge that we have. The first one is waste and bioconversion. Okay, we all claim in our website and during the project that we are using waste to rear insects, but in fact, we have the legislation, and according to the legislation, we can only use the what it is already, at least in Europe, what it is already allowed for the other animals. And that means that we are competing for the substrate, we are competing for the raw material, for the feed, for insects with the other animals. And also, having different waste will change the diet we are delivering to the insects. And this can have an impact on the insect performance, on the product quality, which can, you know, make it difficult to apply in animal feed. Another issue using waste and mixing different kind of waste together is the substrate texture, which is also very important. Not all the substrate will retain the water in the same way, will have the same texture, and the insects will behave differently, you know, with different texture, which is very important. It has been a little bit underestimated sometimes. Another issue is the quantities and the consistency of the production. So this is the more or less the same picture I showed you at the beginning. 2023, we had 1.3 billion tons of feed that were produced. If we look only at poultry, pigs, and aquaculture, it means around 900 million tons of feed that has been produced. So look at only including 1%, which is not an ingredient, it's an additive, in the animal feed that we need about 9.3 million tons. What about the production? Oh, well, it is very difficult to have data on the volume of production, real data, and this is a data I found from Thierry Duvanel of the Buller Group that gave a lecture on the beginning of this year, and he said that the market nowadays is around 0.08, 0.22 billion ton. So definitely very far from the one that we need, even for 1%. And we expect to have something like 3 million tons by 2030, according to EPIF, even if this data have been a little bit, how to say, changed recently. But still, even with 3 million tons, we are definitely still too far from what we need. And the other issue is the consistency of the production. We have seen that in the market, when you analyze different insect meal coming from different producers, you have we have a difference in composition. And if I'm a feed producer and I want to produce and to invest money on the production of a line of product containing insect meal, I need to have, you know, a consistent production, because I need to receive my black soldier fly or my Tenebrio that still have the same level of protein, the same level of lipid. Otherwise, I will have to change, too often my formulation, and also the product that I will deliver to my clients will not be always the same. So quantity and consistency. Then another issue that often many startups face is the scaling up. There are very huge difference between what we do at research scale. We are doing a lot of nice things with very small, and even in climatic rooms, the picture here with the blue trails at my climatic rooms. Also, we are doing the research in I will not say big scale, but still quite nice scale for us at least. But then what about going up a business, upscaling, producing ton of insects? This is not that easy to do, and also the results that we obtain at research scale are not always transferable to the large scale. And for example, well, I have read somewhere that the pilot, and at least the large majority of industry, not industry, of pilot plan are around 50 to 500 square meters. But then to be profitable, you have to definitely have bigger plans, which is not that easy to do. And finally, my last, challenge is the price. Unfortunately, nowadays we still have a too high price if we compare to other commodities. If we want to enter the market of feed as an ingredient, we enter in the market of commodities, okay? So we have to compare with soybean meal, fish meal, pea protein concentrate, etc., etc. But let's try to take the positive things from that, is that we can consider that the industry, it is too small, still too small. There is a huge space for improving. If you see the data of the on the quantity of feed that we need, and you see that there is huge space to increase. And we expect that with the increase of the industry, the price will decrease. Nowadays, I would say the preferred market for the insect products is the pet food. And the pet food is still a small market, but that it is able to pay a lot. So why should, if I'm an insect producer, why should I sell my stuff to a poultry plant when I can sell it to my pet food with a high value? But then in the future, the market will be aquaculture, which is definitely bigger than the pet food, and also poultry and pigs. So once again, this can be that can help the increment, the increase of the production. And then what we can also target is to use insect oil not as an energy source, but to use it as a more valuable ingredient. So to have this kind of very nice impact on the animal health, and for example, in ruminants, in the methane emission. So the idea could be, at least for the moment, to use the insect more as a functional ingredient rather than a commodity. This is always, for the moment, with the volume that we have, they are not a commodity. We still have some too small quantities. So I have still 1 minute left. And so just 1 minute to give you this information. You already know it. But in 2026, it's 1.5 years from now, I will held in Torino the Insect to Feed the World conference. You know that the last edition was this year in June in Singapore. It was nice, it was amazing. So I decided to propose Torino as to host this conference. I have to say that I already feel the pressure on that. So I really hope to see you there. And I will definitely need your help to have a very, very nice conference. Torino is an amazing city to visit. So it can be also a very nice opportunity to take this chance to visit Torino. And of course, I will be very happy to answer any of your questions.
Thank you very much, Laura, for a very comprehensive and fascinating talk. I really found the most interesting thing, which I didn't knew actually, was your remarks regarding the ruminants and the effect of modulating the CH4 emissions. It's really interesting.
Yeah, and there is a very big interest from the feed companies for that. The ones that are working with ruminants are very interested in that because, well, the price is quite similar to other lipids that they put into ruminant feed and can have a very nice impact. Without, of course, also taking into consideration that if they are using cocoa oil, for example, in ruminants, they use it. They take it from very, very far away. So, once again, this is a question of sustainability. You can produce it locally.
Yeah. Okay, so I will leave room for others to ask questions.
I hope to be able to answer. No questions?
Okay, so in the meantime, I have one. You mentioned the maximal inclusion levels of insect protein inside the meal.
Mm-hmm.
Do you think it's due to the chitin as an ingredient inside the meal and its effect on the digestibility?
Well, these are levels, for example, if I'm thinking about poultry, it has been seen that if you go level up higher than 10-12%, some impact on the gut health has been shown. So some some impact on the villi, on the mucosa, intestinal mucosa, that are not that positive. We are still trying to understand if those impacts were due to the level of insect meal or the type of insect meal. So this is something that has to be investigated. Concerning fish, or 25–30%, all the data that we analyze show that up to that level it works perfectly. We also have some trials with higher level. For example, we tested a 40% of inclusion of Hermetia in rainbow trout without any negative effect even on the performance and also on the product quality. So it was also okay. But when we did the research, the review, we really investigated, I don't remember how many papers, and these were the average value. But it always depends on the kind of insect meal that you have, on the level of protein on your insect meal, on the composition, on the process. So there are still many parameters that can impact this level of inclusion. But those are the recommended ones.
I also have a question.
Hello, Laura.
Hi, my name. I'm from IGA, the Research Institute, working on the BSF. And in our consortium, as you probably know, Idan, I'm sure told you, we pay a lot of attention and time and research trying to figure out the exact composition of wastes that would make the best black soldier fly. How much is that a central research question as far as you know? And how much impact does it really have on the nutritional values of the black soldier fly? Like we really try to go into detail of what % of tomatoes and what % of corn waste or banana waste would be the best black soldier fly. But is it really a real question, or as far as you know, it's not that important?
Well, I think that this is very important because I think that not all the waste will be digested in the same way by the insects, and also when you are combining the waste together, they could change also the digestibility because there is a combination. And then changing the composition of your substrate for your insects, it will change the global digestibility, then it will impact the performance, and it will impact also the composition of your larva. So I think personally, I think that it's important because we have seen doing research that even maintaining the same nutritional composition in terms of protein, lipid, energy that we deliver to the insects. So we formulate diet that are ISO in all the terms. We have different results because of the different mixtures. We are using different substrates. We are combining them in different ways, still keeping all the diets the same from a nutritional composition. And we have huge differences in terms of performance and in terms of impact. For example, in the Advagromet project, well, in our case, we worked with Tenebrio, and Hermetia was for another partner. But for Tenebrio, what we did is that we prepared 6 different diets that still had the same nutritional composition. We mixed a different substrate, and then we had, for example, we had only 3 diets that were really working. And among those 3, we selected one diet that gave us the best results, and we push it in mass production for the production of larvae, of live larvae for poultry. So yes, it has an impact, definitely.
Okay, thanks. If there's no other question, I have another one, but maybe someone else would like to ask.
Yeah, ask it.
Okay, so another thing that interests me, looking at as a black soldier fly as fish meal, do you know of any research that is done on comparing the composition for cold water fish as warm water fish? Did you see much differences, like in trouts or salmonids for the cold water and like barramundi and denise or whatever for the warm water?
Yeah, there are differences because, well, the fish are quite different. So the physiology of the fish is different. For example, if I'm comparing marine species and freshwater species, we have seen that with freshwater species, they are able to better use it. I don't know why, but then if compared to marine species, sometimes with marine species, you cannot push the inclusion level that high. And I think that it's also happened with warm water and fresh water. You know, there are differences, but there are, and it is always difficult also because maybe 2 research have been done with yellow catfish and rainbow trout, for example. But then, and maybe they both use black soda fly meal, but then the black soda fly meal was different. So it is also difficult to compare those results because the insect meal at the beginning, it was not the same. So it's still something that is not, but I would say that for all species there, it works.
Okay, thank you.
Heinrich, you had a question?
Yes, I do. They're my favorite topic, the animal welfare. As you know, I'm pleading for a 6th category of animals in the European Union. Right now, there are the 5, the farmed animal, the pet animal, zoo animal, fur animal, and wild animal. And insects are under farmed animal, and that does not fit.
Yeah, I agree.
So, okay, maybe a long fight. However, if we let's assume that we have this category, I understood from your talk, one is the killing method, which has to be described. But what else should we focus on in animal welfare? In what topics? Your other example, yeah, if you look for pig, of course, it's obvious to have a certain amount of pigs on a certain space. It's counterproductive at like soldier fly, for example. So Chad, do you have an idea what kind of animal welfare parameters we should look for besides killing?
Oh, this is a very difficult. I think, well, maybe density, but we are still very far away of having, because I think that the density for a black soda fly would be completely different from the one of a cricket, for example. So yeah, density should be investigated. Temperature, maybe there are also some, you know, temperature where they are feeling better rather than in other. You know, sometimes maybe for some welfare people, they will say, oh, it is too high temperature. But if you put them in a too low temperature, it's not a good thing. So likely also temperature. And I think that there are, well, I don't investigate those aspects, but there are some thermal camera that can be used to see also the behavior of the insects linked to the temperature. Well, and then killing methodology, but yes, but killing methodology, I would say the one that killed them rapidly.
Yeah. Okay.
So it's a big issue, you know, because until yesterday we were not caring about that, and I agree with you, Erich, that we should have insect outside the animal farm, the farm and animal legislation, because this would make much more sense for insects, also for the substrate that we can use and for the bioconversion that they are able to do. But then this is it will be a long fight, but maybe if we continue to fight.
Yeah, we never give in. Thanks for the support. Okay.
Okay, I think we ran out of time, so
maybe there is something in the chat. Oh, no.
Yeah. Let's see.
Maybe perhaps stress, yes, stress in insects, yes, but how to measure the stress? Which are the parameters that we are measuring in the insects? You know, already the parameters, the stress parameter that you can take, for example, from the blood in other animals, it has been seen that they are not that reliable, because when you are collecting blood in the poultry, it will stress just because you are collecting the blood. So it is some. There are now some parameters, for example, that we are investigating in poultry is the cortisol in the feces, to see the level of cortisol, and in that case, there is no stress because you are collecting the feces. But then for insects, I don't know. But yes, this could be something that could be investigated.
Okay, Laura, I think I have at least 3 more questions, but we ran out of time. So I really appreciate your talk, and thank you very much again.
You're welcome.
And we're looking forward to see you in 2026 in IFW, of course.
Yes, definitely. But I will contact you before.
Of course.
Okay, so thank you very much for inviting me once again. It has been really nice. And please, if you have any question, if you want more information, don't hesitate to write me. Okay?
Thank you very much.
Thank you very much.
You are welcome.